Okay, and now give me just 1 second to get the next thing queued up. Okay, so we're gonna bam. Okay, so note, when we first started to talk about PCA, I mentioned that we were going to talk about it from the perspective of something called singular value decomposition. Oops. Singular value decomposition is the standard way of doing PCA. However, there's an older technique that gives the exact same results based on correlations among the data. So here, I just want to give you a little intuition of how PCA can be thought of in terms of a transformation of these correlations. So with that said, let's say we had some normal cells. These are just, cells from, biological cells. I don't know where they're from, but they're just normal. Psst, if you're not a biologist, these could be people or cars or cities, etc. It doesn't really matter. So even though they look the same, we suspect there are differences. These might be one type of cell, or one type of person, or car, or city, etc. And these might be another type of cell, and these might be a third type of cell. Unfortunately, we can't observe the differences from the outside. So we sequence the messenger RNA in each cell to identify which genes are active. This tells us what the cell is doing. And if these were people, we could measure their height, blood pressure, reading level, etc. Anyways, here's the data. Each column shows how much each gene is transcribed in each cell. Boop, 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 boop. Now, let's imagine, just for now, there are only 2 cells. We can have, when we just have 2 cells, we can plot the measurements for each gene. This gene, gene 1, is highly transcribed in cell 1 and lowly transcribed in cell 2. So notice this graph is different than what we had before, where we were plotting the genes were the axes. Now we're plotting all the genes on axes that are for the cells. So that's one difference in perspective when we're talking about correlations and doing PCA versus SVD, or singular value decomposition. That's the primary difference. Okay, and this gene, gene 9, is lowly transcribed in cell 1 and highly transcribed in cell 2. In general, cell 1 and cell 2 have an inverse correlation. That means that they are probably 2 different types of cells, since they are using different genes differently. Now imagine there are 3 cells. We've already seen how we can plot the first 2 cells and see how closely they are related. Now we can compare cell 1 to cell 3. And we see that cell 1 and cell 3 are positively correlated, suggesting they are doing similar things. That means the same genes that are doing the same gene doing a little amount of work in cell 1 is also just doing a little amount of work in cell 3. Meanwhile, out here on the upper right-hand corner, we've got a gene that's highly expressed in cell 1 and highly expressed in cell 3. So these, in both cells, the genes are kind of doing the same thing. Lastly, we can also compare cell 2 and cell 3. And the negative correlation suggests that cell 2 is doing something different from cell 3. Alternatively, we could try to plot all 3 cells at once in a 3-dimensional graph. So here we've got cell 1 on the y-axis, cell 2 on the x-axis, and cell 3 on the z-axis. Cell 1 could be the vertical, so like I was just talking about. And again, cell 3 is that depth thing, which is kind of hard to imagine, but we're just going to go with it. Okay, we could then rotate the graph around to see how the cells are related to each other. But what do we do when we have 4 or more cells? Draw tons and tons of 2-cell plots and try to make sense of their correlations? Or do we draw some crazy graph that has an axis for each cell and makes our brain explode? No, both of those options are just plain silly. Instead, we draw a principal component analysis graph. So what we've been talking about all along. A PCA converts the correlations, or lack thereof, among the cells in a two-dimensional graph. Cells that are highly correlated cluster together. So this cluster of cells are highly correlated with each other. And so are these. And so are these. To make the clusters easier to see, we could color code them. And once we've identified the clusters in the PCA plot, we can go back to the original cells and see that they represent 3 different types of cells doing 3 different things with their genes. So in this case, we've used PCA as a way to sort of identify sort of unique subsets from the original data. So we just had a bunch of measurements from a bunch of cells, and we didn't really know how to classify those cells. And we used PCA as a way to kind of see if it can make sense, help us make sense of the data. And it did that by compressing the data into a 2-dimensional PCA plot. And in this case, it focused more on using the correlations rather than sort of finding that best fitting line. And I'm not a huge fan of using this approach, but people still do teach this way. And when you see PCA out in the wild, they'll say, well, we use the covariances or something like that. And you go, okay, that's fine. That's like SV, singular value decomposition, but harder to understand. But the results are identical. Okay, so bam. Here's one last main idea about how to interpret PCA plots. This is stuff we've already talked about. The axes are in order of importance, and the more important differences along the second principal axis. Yeah. Okay. Boom, boom, boom. So I'm just sort of repeating what we're doing. So what I'm going to do is I'm going to pause the video because we've pretty much got PCA down solid right now.